Hi everybody, thank you so much for coming. For those of you that don't know, my name is Ramya and I'm the Director of Environmental Science at the Key Biscayne Community Foundation. Every last Thursday of the month we have a citizen science lecture for you. Sometimes I'm the lecturer myself, sometimes I bring guests. You know, we always cater these lunches for you guys because we really appreciate you all coming. Now we have Milanesa who will be here a little bit coming. Today for our lecture we have Anastasiya Plotnikova. She is a PhD student at the University of Miami at the Marine campus right over on Virginia Key. Anastasiya and I actually worked in the same lab for several years. I also worked with Toadfish. They are fun little guys. They're angry little fish. We like to refer to them as the Oscar the Grouch of the sea. They're ugly, but they're kind of so ugly that they're cute. She's going to go into why we use toadfish and why they make a good study subject, and all the different things that we have learned from them and how they've actually helped us in a lot of ways to find out a lot of things that are going on in our bay, and how they helped us get rid of the Ultra Music Festival that they brought out to Virginia Key for 1 year and then didn't bring it back. The toadfish played a part in that. So without further ado, we'll give it over to Anastasiya.
Hi everyone, my name is Anastasiya, as Ramya introduced me. So nice to be here. Thank you for listening to me. We're going to make this fun. We're going to talk about fish. This is my favorite type of fish, the toadfish. Have any of you guys heard of the toadfish ever before? Yeah, a few people, maybe? You've caught them. So if you've caught them, you must know that they are actually local, right? So that's what they look like right here. They are actually scaleless. As you could see, there was like kind of a sheen on them, which is pretty unique. And that's kind of the reason why they're called toadfish, because they look like toads, they make a toad-like noise. It is hypothesized they can actually breathe through their skin, which makes them really suitable for living in Biscayne Bay that experiences these nightly low oxygen and low water, right? Like when low tide comes in, right? So they are able to survive in that just fine. You said you caught them, right? So you dived a lot. They love to hide, right? They are ambush predators, so they do not like to be out in the open. They're not pelagic tunas or mahi that you think about, right? They just kind of like to bury down in the sand, and their coloring allows them to kind of do that really well, right? They camouflage and you can't see them. We had one of our professors in the department go out and snorkel as well, and he got a little video of them. Actually, recently he also saw a pair breeding out in the wild, so they were actually creating a little nest in a glass cup, like just a glass cup out in the bay, in the turtle grass and the seagrass. Something surprising about them is that their number one predator is actually the bottlenose dolphin. And they like to munch on shrimp and squid, which is what we feed them. And they're pretty hardy animals, which makes them great for studying their physiology and how they respond to environmental stressors, which is what we do, basically.
How big are they?
Great question. So they don't get much bigger than like this. Yeah. Like biggest probably like 2 feet. They have little teeth, but a very powerful jaw. But they're not like super crazy teeth. Yeah, just very powerful jaw. What do we do in the lab with them, right? They're great model organisms for kind of understanding how pollutants, right? There's various pollutants out in the bay, and how those impact them. We use them as model organisms because they keep well in the lab. All you need to keep them in the lab, we have a whole room full of tanks, probably like, I don't know, Ramya, like 50, 60 tanks, right? And all you need is some seawater. We get it right from the bay. It goes through a little filtration, an air stone, and a few little PVC pipes that we keep as shelters for them so they don't, so they kind of get, a little area to hide. They don't like to be super social, as we mentioned. So the males are actually very territorial. Because they are the ones that guard the eggs once they are laid by the female and once the male fertilizes them, which is pretty nice, right? It's refreshing. The male has to take care of the young and the female just goes off to do whatever.
Is this the kind of fish you would catch as sort of ornamental?
No. No. No, not for eating. That's a great question. She asked, what was your name? Olga. Olga asked if this is a type of fish you would find on a menu. No, you would not find it on a menu. It's pretty gross.
I've never seen it, so
Yeah.
that's what I was
It's a great question. Because they're benthic, they don't have a lot of muscle mass, so there's nothing to eat on them. I've dissected a lot of them, and trust me, there's nothing to take home or to even pick at. I have a little video for you. You said you caught them, right? So you dived a lot. And is it easy to find them? Is it easy to spot them, do you think?
Yes, I don't. The other thing, sometimes they hide underneath the
Yeah,
grass.
you're exactly right, right? They love to hide, right? They are ambush predators, so they do not like to be out in the open. They're not pelagic tunas or mahi that you think about, right? They just kind of like to bury down in the sand, and their coloring allows them to kind of do that really well, right? They camouflage and you can't see them. So it's actually pretty hard to spot them. Oh, there we go. Yeah. That's the noise that they make. Do you guys see where it is? It's right there, in that little crevice. Right there. See that? There's the eye. The mouth is opening. Other eye. Pretty neat, right? So it would be kind of hard to spot this, right? Out in the wild. They're pretty well camouflaged due to their color, as we can see. Stress response, as many of you know, right? What are some things that cause stress in your life? I asked this. Traffic. That was one of mine. Oh, that was one of my bullet points. Perfect. Traffic. What else?
The computer.
Computer. Yeah, that's one of my stressors right now. Lack of food. Yeah. Maybe relationships, right? People. And so what kind of stressors along those lines do you think toadfish experience? Do you think they experience traffic as a stressor? Not particularly. Perhaps boat traffic, right? Like noise from the boats. Predators, right? Exactly. Bottlenose dolphin. You hear them coming. They experience stressors like other toadfish, predators, right? Lack of food, stuff like that. I give lots of talks to little kids, actually, and I love doing that because they are so curious. And oftentimes I'll bring in, for an elementary school or a middle school, I'll bring in a tank with fish. And they get to touch it. So I take it out of water. Because like I said, they can stay out of water for a super long time because they can breathe through their skin. So the kids love them. Okay, perfect. So this is a stress response I was talking about, right? The things that cause you stress, traffic, relationships, etc. This is kind of the general human physiology outline, right? The pituitary gland secretes this hormone that then triggers the adrenal glands to secrete cortisol. We've heard of cortisol, I'm sure, right? Increased cortisol means that you're in a stress response. And also adrenaline, which is kind of like the more immediate thing that happens to you that will increase your heart rate, make your breathing go faster, right? And increase sweating, like clammy hands, like I have right now, like all that, right? And so these are the kind of things that we can measure in toadfish, actually. So what we can do is take their liver and see how much glycogen it has left and use that as a measure of, well, how much stress did it experience, right? Because how much glycogen is left and then how much glucose is in the blood, we can do that. We can also take cortisol samples from the blood, which is one thing that we did, and I'll talk about that in a bit. And what I do right now for my research is take a look at the heart and how it responds to specifically pharmaceuticals. So what I do is this heart surgery, and I'll literally pump this drug into the heart and see what happens to heart rate and stroke volume and cardiac output, those heart function parameters. Why study the toadfish? So they are very similar to us. They have cortisol, as I mentioned, just as humans do. And these are the stressors that we talked about earlier: traffic, work, relationships. The toadfish have stressors like low oxygen, pharmaceutical pollutants, and the Ultra Music Festival, like Ramya mentioned. Okay, and then the overarching theme of all those things that are stressing out the toadfish is humans, right? Those are all anthropogenic stressors that we've created. Okay, so this is a picture of beautiful Biscayne Bay, right? You can probably recognize it right here. But as we mentioned, it is home to a lot of bad things. So one of those is hypoxia, low oxygen. I'm sure many of you guys have heard about the fish kill that happened in 2020, right? Maybe you've seen this graphic visual, right? Lots of dead fish in a short span of time. And that is due to eutrophication. And then this is a graphic from Miami Waterkeeper that kind of lists the species affected that they could identify, right? You have some pufferfish, sand diver, a lot of unidentified, and a lot are the Gulf toadfish, 44.8%, right? Which brings them into this scope, right? Makes them super relevant to understanding why. And this is actually really interesting because I just said at the beginning that they're pretty hardy fish, right? I said that they're pretty good at responding to low oxygen, right? They can breathe with their skin. I take them out and show them to little kids, right? Well, what happened here? That's like a big point of research that's going on. Some people over at Rosenstiel have hypothesized that it could be what's in the sediment. Because as we mentioned, toadfish are benthic, right? So it could be a buildup of something in the sediment.
She was mentioning, of course, how toadfish are very hardy fish, and they deal very well with stressors like low oxygen. They breathe through their skin, all of that. One of the things that we do, experiments that we do, involves doing small operations on them and putting in wires and things like that to measure stress responses and various things. So to do that, we have to put them under anesthesia and take them out. They're fine for a surprisingly long time out of the water, and sometimes if they're out long enough, they will wake up while you're doing that because they can breathe through their skin. So it doesn't matter that they're out of water. If you're taking too long to get your surgery done, they will wake up and they'll bite you. Thank you, Ramya.
Have you ever been bitten?
Yeah, I have never been bitten. I say that and watch me get bitten tomorrow. No, but I've heard that it's kind of painful.
It feels like being bitten by sandpaper.
Okay, yeah, not terrible.
It's annoying.
No sharp teeth. I don't know who, you were asking about teeth, right? Yeah, no, not super painful. Okay, thank you, Ramya. So the fish kill, how does this happen? I'm sure many of you are aware it's a combination of increased nutrients, right? That may come from septic leaks, also from a fertilizer, right? Like those nitrates and phosphates that we put into our lawn and then get washed off with rainwater, like to go into the bay. Stormwater, all that contributes to an increased source of nutrients for this algae to grow, right? That produces algal blooms. On the West Coast, we have the red tide. Here we just have the, thankfully we don't have that here, but we'd have the regular green algae blooms. And what happens then is that algae needs to decompose, right? And there's such a huge volume of it, that process of decomposition takes up a lot of oxygen from the water. And that leads to the fish not having enough water. That's basically how that happens. Okay, so this is the fun part. So Ultra, as Ramya mentioned, Ultra was held right at Virginia Key Beach a few years ago. And so one of our people in our lab actually, Dr. Maria Cardellano, I knew her when she was a PhD student, she brought pools, little kiddie pools, right, inflatable, with seawater, out right in front of the Rosenstiel School, which is really just across the street from where the festival was being held, put a few toadfish in there, and then took blood samples to see how much cortisol they had, before, during, and after, right? And so this is from EDM.com. They had, the phrase "drop the bass" had taken on a new meaning. I thought this was pretty funny. They have this toadfish right in front of the crowd. And then this is from the Miami Herald, I think, Ultra Music Fest sounds stressed out nearby fish. And this is based on that study. And here is just a visual. This is what she had in her paper that she published along with other scientists here. So this is what the visual, right? We have these two poles that I was talking about. And then she measured the decibel level, like the frequency, in the air and also underwater, right? Because these guys live underwater, so that's relevant to not only them, but other fish, right? Other animals in the area. Oh yeah, they can hear.
I mean, do they have a hole in there?
Can't see it. Yeah, can't see it, but they definitely can hear.
I mean, they hear the music or the vibrations.
They hear the music. They can feel the vibrations as well. Yeah. Yeah. Anybody can hear the music. It's so loud, right? And so this is a graphical representation of that cortisol I was talking about. And they did 3 weeks before ultra, and then 4 days before, and then night 1 during ultra, right? So you can see that B right there is different from those A's, so that's significantly increased compared to previous. Okay, and this is pharmaceutical pollution, the other stressor that I was talking about earlier. So this is a study out of FIU. This is just the headline of a newspaper, but it's based on a study out of FIU where they tested fish, and they found that there was, I think, 16 different types of pharmaceuticals just in one fish, which is pretty insane, right? And we're talking about like antidepressants, beta blockers, right? And then this is another paper, Understanding the Occurrence and Distribution of Emerging Pollutants and Endocrine Disruptors in these Sensitive South Florida Ecosystems. So the reason why we have them is because not all of the kind of the active ingredients that you take in right through your prescriptions are going to be broken down completely by your body. And that's going to be in the wastewater, right? And wastewater treatment plants aren't exactly up to speed with the sheer amount, right, of pharmaceuticals that are present, especially now. Also, of course, you have people flushing pills down the toilet, right, and all that kind of contributes. So this is why it's showing up in the water. Yeah. No, we get our water supply from, I think, the Biscayne Aquifer, I think, Ramya, right? But we don't get it. This is like the bay. This is like salt water.
So this area specifically was what I studied when I was in the lab. We do get our water from the aquifer, not from the bay, so it is much cleaner. I'm not going to say how clean it is, but it is also filtered. It is much cleaner than what's in the bay, and it's not what we don't drink bay water. But I mean, personally, I don't think any of it's clean enough. But as far as how it affects humans, we don't actually know. This area of study is only about 20 to 25 years old, which seems like a lot, but that's actually for long-term studies and effects, we don't know, because 20 years is not really, the length of a human life, so we don't know what kind of effects it has on people. For the most part, the amounts of pharmaceuticals are low enough that we're pretty certain that there's very little to no effect on people. But even these low amounts of pharmaceuticals in the water can have an effect and do have an effect on the animals that live there. But again, as I said, there are no long-term studies yet. They're being done, but we don't know yet. So as far as, is it a cumulative effect over time? Do these small amounts still have an effect on you in some way, even if they're not accumulating? We don't know yet, but that's something that they're working on.
I work on heart stuff, right? So I expose them to pharmaceuticals, specifically an antidepressant. This is fluoxetine, which is the main active ingredient in Prozac. And what happens in the brain and when you're taking this antidepressant, right, is between these neurons, right, if this is one neuron and this is the beginning of another neuron, you have this synapse, and serotonin is going to be these little red dots, it's going to be released, right, into the synapse. And then to terminate that signal, you have to take it back up into this first neuron. But what happens with the SSRI, which is like the fluoxetine that I talked about, is that it blocks this ability for it to reabsorb. So essentially, in the end, there's a lot more red dots, serotonin in that synapse, which is what makes you happier, right? But serotonin is involved in a lot of other things in the body, like heart function, lung function. There's a lot of it in your gut. That's the main site where it's produced. It's involved in digestion. It's all over the body. In mammals, there's over 15 different subtypes, receptors, and receptors are the things that will take in that serotonin and mediate a response from a cell, right? And then that cell will mediate a response of the organ, and then that organ affects your other organs and all that. And so for toadfish, this is relevant because this is heart rate on this axis, and this is time. This little gray area is hypoxia, so that low oxygen stage, right? Normally, to survive that hypoxia, what they will do is go into a little hibernation. They will lower their heart rate. They're going to a bradycardia. That's this dark black line with the black circles. But if we give them an inhibitor of one of these receptors, which is these two lines up here, that bradycardia, or low heart rate, is not going to happen. It's going to be attenuated, right? So that's bad for them and their survival, right? Because if they're not able to do what they normally do to survive low oxygen, that's an issue, right? I think we are about done. This is the last slide. If you have any more questions or comments, if you want to talk to me, like I said, I love talking about this stuff. This is my email, and this is my supervisor's email, of course. Thank you so much for having me, and I'll take questions, yeah.
I just wondered, do you ever give tours of the,
Yeah, we give lots of tours. Yeah, I think you have to go through kind of the outreach office, but yeah, you can email me if you'd like, and I can connect you with them. Yeah.
Was there any other biological reasons to get ultra moved away?
Oh, a lot. I know the toadfish angle. I don't know about other fish, but.
So I'm sure she talked about, I missed it, but I'm sure she talked about the toadfish response, their stress response to ultra from the sound. Their response was, did you mention, equivalent to, their stress response to the sound coming from ultra was equivalent to the stress response they have by being chased by a predator. So that's how much it really, really stressed them out. That was the only fish that that was tested at the time. But it was tested in the hatchery, which is across the street from the University of Miami Marine School. And the hatchery is full of all kinds of different species of fish. And even though they weren't tested, a lot of them are like much more fragile, or they just don't handle stress quite as well as a toadfish does. So we can only imagine how that affected them. But the other reason, obviously, is that a good part of Virginia Key is a wildlife preserve. And there's birds that nest there, and a lot of birds that come through periodically as they're migrating. And this was right during part of migration season for some of the birds, so that was a concern because the noise obviously would scare them away. And just to preserve, we didn't know what was going to happen with all of these, literally hundreds of thousands of people in that area making a lot of sound, possibly causing a lot of pollution. How is that going to affect the wildlife preserve? I will say that the producers of ultra did a pretty good job of containing it in the area that they were designated, so people didn't really get escape from there and cause problems. But it was enough of a concern that it was too much of a hassle for them to try to do it again, and they got a lot of bad press internationally from the toadfish study.
do you do any work with Nova across the street from you all?
No, not our lab specifically. Other labs within the department do.
One of the other experiments that they do in the lab is taking fish and putting them in what we call a swim tunnel. And basically you take a single fish, you put it in this tunnel, and it pumps water through. And you can change the speed that the water is going through. And fish automatically, when they're faced with some kind of current, they face into the current and they start swimming against it. And that way we can measure different things like their respiration, how much energy they're using, how much stress it's causing them, all those kinds of things. And, then we can slowly like take up the speed of the current, and then, they'll swim faster. They'll change the way that they're swimming. If it's like a leisurely swim, they're kind of like this. The faster the water, they'll tuck their side fins in, and they're just going straight and like, just moving their tail. So, there's all these different like behavioral things that they do. When you put a toadfish in a swim tunnel, they are super lazy, and they don't want to swim. They like to just sit on the bottom. So you turn the speed up at all, and they just go flat against the back of the swim tunnel. It's really, really hard to measure these things with them because they just refuse to swim. So they are a good test subject for a lot of reasons, but they can be very difficult test subjects.
What's their lifespan? We are not sure exactly, but probably about 8-9 years. Yeah.
I think each female will only breed once per season, per breeding season. There's usually, 2 breeding seasons in a year. They just started a breeding program in the lab so that we don't have to use wild-caught. We still use some wild-caught toadfish, but we've been breeding them now for a few years. And so it also helps because now we can do experiments on the eggs, on the little larvae, on like the different stages of life. And so we can get a more complete view of how this, particular animal is affected by all the different stressors. So instead of just seeing how like the adults are affected, now we can see it more completely throughout their whole lifespan. But yeah, so usually once around the springtime, and then again like several months later towards the fall. And then the number of eggs that they lay is kind of based on the maturity of the female. So in the lab, our females are generally about that big, maybe 6-7 inches at most. And we have these PVC pipes that they'll go inside, and you'll have a couple, a male and female, that'll decide, okay, this is the PVC pipe that we're going to use for our house, and they'll lay the eggs in there, and then the female leaves. She just takes off. The male stays in the pipe and protects the eggs and fans them with his fins so that they get oxygen. And he'll stay there until they hatch. And they'll hatch, and initially they stay stuck to the side for a while, and then eventually they become free swimming, and as they start leaving, then the male will leave. We found, I think it was Mike Shimali, the professor that gave us that video in the beginning that had the boat whistle sound that they made. He was diving, and he discovered, this was the year before we started our own breeding program, he discovered a fairly large PVC pipe in the bay, and it was about this big, and it had a monstrous, huge toadfish in it. And inside, the entire inside was lined with eggs. And he brought it back to the lab, they hatched in the lab, and we got, I think, 3,400 eggs. So 3,400 babies, or 3,200. We split them between the 2 labs. There's another lab that also uses toadfish. So each lab got 1,600 little baby toadfish from that. But that was like, a very mature, very old, like much older female. And so the bigger they are and the older they are, the more mature they are, the more eggs that they can produce. So the females we have in the lab produce anywhere from like 20 to 100 eggs. I think, something like that.
We've had 200.
Oh, okay, apparently up to 200. But obviously nothing in the neighborhood of 3,200. So very few of them. Just like most animals, especially when they're produced in large numbers, they're produced that way because they have a lot of predators. When they're really small. So very few of them will get to adulthood.
That's why you have a lot of eggs, I guess.
Yeah, exactly. That's why turtles do it too. You have 1 in 1,000 turtles will make it to an adulthood, but that's why you have over 100 eggs in every nest, pretty much. Yes?
What is the worldwide distribution of the species?
Well, this Gulf toadfish are just around Florida and in the Gulf of Mexico. They're named for the Gulf of Mexico, that's why they're the Gulf toadfish. But there are various species of toadfish that are found worldwide, and some at various depths, like some of them are actually fairly deep. But they can look really different from each other. It was actually, we were kind of laughing during the ultra study. One of the news articles that came out had a picture of a toadfish, because that was the study. And it was the completely wrong toadfish. It was a toadfish that lived at like 5,000 feet of depth, that wasn't even on this side of the world. They just like Googled toadfish and used that picture, and it was completely wrong. It was funny. So the question was, are toadfish declining more than other species in the bay? Toadfish are not considered endangered. there's millions of them. So there's not really any concern for that. It was surprising, as she mentioned, that during the hypoxia event that happened that killed off thousands of fish, that a huge chunk of that was toadfish. Part of that is because they don't move around. They like to just sit in one place. And so when you have a hypoxic event, you lose oxygen, they're just sitting there and suffocating, whereas other fish will actually swim away. So that's probably a large reason for that. But we also know that they tend to be very resilient against hypoxia. So it's still, despite the reasoning, it's still kind of a surprising result. But I don't think that at this point there's any concern that they're going to be endangered anytime soon. And there are a lot of other fish that are much more fragile and much more susceptible to these problems. And so when they happen, they're going to be in a worse situation than those fish would be.
To a while back, the master's program is over here, correct? And then you just started.
There's like 3 different tracks. There's the MPS program, which is fairly short, I think like 1 or 2 years, the Masters of Professional Science. And then there's the Masters, and then there's the PhD program. So yes, I think a lot of times students who are kind of unsure of directly what they want to study will start with the MPS track and then transition. You can transition to a Master's or a PhD from there. Yeah. All right. Thank you.